The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 15th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry CRO space for over 13 years.
We also have a special guest with us today. Kelly, would you like to say hi?
Hi, thank you so much for having me.
Yeah, we're excited to talk with you today. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional location in Europe and in Australia, we're both here in Philadelphia, and our producer is in Missouri. So we're truly all over the map. We're amped to have you listening to our 15th podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener at any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're excited to have you with us and look forward to a fun episode today. To kick us off for episode 15, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. I don't want to say that we've got a packed agenda today, but it's definitely quite full. We do have some medical research news, and the theme of today's medical research news, I think, is FDA guidance documents.
Yeah, and I will say I have to thank you there. I know I busted your chops on the last episode about saying we had a packed agenda. We always do, but you went to a different route there, so thank you. And yeah, so guidance. Why is it important? Well, this is the thing that's been coming up a lot. We've heard this in conferences that we've been to. We've talked with clients, and the industry really is moving towards more and more of a framework for how to properly do things, do assays, flow cytometry, how to validate. We've talked in the past about H62 and the guidance for validating flow cytometry-based assays. And so here, what we've got is, and I'm just going to lump them both in together. Number one is the considerations for generating clinical evidence for oncology multi-regional clinical development program. So this is FDA guidance. That was a mouthful. And then number two, developing potential cellular and gene therapy products, right? And so the FDA is putting together some thoughts on how to proceed. Now, Brian, why is this important? Why do we want this information?
As much as people are concerned about regulations and don't want to regulate industries to death, when there are insufficient regulations, it leaves too much sort of gray area and subjectivity as well. So in the case of flow or even cell and gene therapy, frequently we get the question of, is this good enough? And why do you think this is good enough? Or what are the criteria for good enough? And if there's not a really very, very specific guidance on what that means, so it's something like, you must do 3 replicates of this to be enough, or you must have at least 10 subjects, or this widget must be at least 10 centimeters long, then it can be difficult to say this is good enough, and you may be constantly optimizing something that may not even be important. So it makes it very difficult from an industry perspective to satisfy requirements when they're not specific.
Right. And you want to take away the subjectivity of portions that really shouldn't be subjective, right? When we're talking about statistical analysis and number of replicates and things of that nature, there really are equations to be able to determine what is most effective, what should be used there. When clinical trials are designed, how many subjects should you have to make an effective decision? And then, the guidance comes into when you have secondary indications or, various, what was it, when you've got cancers, but then there are subtypes, right? How you should add those to the trial to make sure that you're evaluating appropriately. And I will say, I will even go one further. I don't know if you've ever read the book The Emperor of All Maladies, referring to cancer. It goes through really the history of clinical trials a lot, and what it was like back in the 70s and beforehand, and the lack of guidance, the lack of regulation. I mean, from one study to another, you just couldn't compare. And what we really want to get to is there's so much great work going out there, so much science, and you want to be able to harness all this information. You want to use it for the greater good. And if every study is its own unique entity and cannot be compared to another study, well, then we're all losing out, right? And so that's where this guidance, not only does it help the greater good, right, which is something we're striving for, but it also helps to make sure that these studies go off and are as effective as they can be, so they don't have to be repeated. I mean, let's be frank, clinical studies are not cheap. They're incredibly expensive. They're time consuming. And that's not to say the personal aspect too, right? You've got patients that are submitting samples alongside getting treatments. And so you want to make sure that you're doing right by them as well.
Exactly. The analogy I always use is, do you want to continue throwing pebbles into the ocean, or do you want to start building a bridge out to a specific island? And with these types of draft guidance, ideally we're using these clinical trials to build that bridge instead of the pebble analogy. And so I did also want to call out the fact that these are draft guidance. So to all of our listeners, please make sure that if you have any association with the topics under these draft guidance, to go in and actually submit your responses. The FDA does look carefully at these, and they definitely put higher value on well-supported or sort of germane recommendations. They're there to guide the whole industry, so they're looking for as much feedback as possible. And of course, both of these guidances are directly applicable to flow cytometry, since oncology multi-regional trials with our multi-site global spectral harmonization, this is near and dear to our hearts. Right now, the agency guidance, a part of it that's most frequently cited as unclear in my discussions with sponsors, is what does it mean to have a representative sample from each ethnic group that you are looking for? And sometimes this is sort of applied differently, and clarifying exactly what this means, what regions that they're looking for, and what regions could be considered applicable to the U.S. population would be a huge win for setting up these multi-regional trials. And then from a cell and gene therapy standpoint, there are untold number of questions, but several of them are definitely addressed within this guidance document. And from a flow cytometry perspective, we're looking for guidelines on the analytical rigor required at different steps of the clinical trial process, what to do with very rare samples, and how to validate things that may not be there, which coincidentally are some of the workshops that we submitted to Cyto. So hopefully a timely thing there as well.
I mean, one of the very interesting things that I found from the multi-regional clinical trial thing was the understanding that standard of care is not the same across the world, right? So the FDA guidance is obviously for the US here. And so there is a specific standard of care. Well, I should say, they even do address that across the U.S. there may be variations of standard of care, but then talking about what you should do if you're having a global trial and there's standard of care one place and you want to include controls there, you should have controls for the US. It kind of makes sense that you're representing all the populations, but it's good to make sure that that is addressed up front. The other thing too, of course, I go to meetings here, right? What do we spend a lot of time in meetings? The cellular and gene therapy products one talks about interact, right? And I went through and I was looking up a lot of the acronyms and what do they mean. And so really talking about early meetings with the FDA to answer questions, get info on novel products so that you're setting yourself up for success, right? The FDA obviously is invested in not wasting time as well, and so have set up meetings to be able to answer these questions and keep things moving. You mentioned about regulation, so on and so forth, but in this case, I think very well intended there. And two, it's got a lot of information as to things if you're curious about the process. What is an IND, an investigational new drug, right? What is a BLA, a biologics license application, and kind of what those are in process here to get to the endpoint that you're looking for.
Yeah, and a lot of these things are frequently locked up behind sort of proprietary processes or consulting agreements or other things. So I think getting that out into the public sphere as soon as possible and just letting them know that this is an avenue that's possible is always a strong positive there as well, especially with these cell and gene therapies that are frequently targeted towards very rare populations and are being sort of developed as quickly and efficiently as possible there.
Yeah, and I was tangentially aware of a lot of this. My previous roles at a company like Janssen certainly was present in teams there, but I was in discovery much more, so didn't have my hands on it. And going through these documents, it's informative, right? You get that roadmap, and so highly recommend if you're interested in that area, you're going beyond discovery and into more clinical development, more later development. You'll learn a lot there.
Yeah, and even better, ideally, you'll be able to find new questions to ask the FDA, and so please do pose those and make sure to send those in.
Mm-hmm.
Yeah. So that's the medical research news for today. The other news we've got is, of course, investment news. And here I've picked two stories on Gilead with a new ADC molecule and then Incyte with a new monoclonal.
Yeah, and so the big story is really, and it mentions the comeback of Gilead, right? And so what's the comebacks? They had Trodelvy, had failed in a couple of trials, phase 3 for bladder cancer and non-small cell lung cancer, and so looking to get back into the ADC space. So antibody drug conjugate, so binding, conjugating a payload to an antibody, using the antibody to then target to where the therapy needs to go. And in this case, the company that they're partnered with, Tubulus, is known for their linker technology, specifically P5 conjugation, that's supposed to reduce the loss of payload before hitting target. On their website, they mentioned as much as 60% of payload being lost for other conjugation technologies. And so what does this do? It essentially allows for fewer off-target effects, lower toxicity, and so definitely interesting to see how this continues to go.
Absolutely, and definitely the ADC space is very, very popular right now as people are seeing the efficacy of these new molecules. A lot of them are being commercialized, and they're definitely at the center of a lot of the biotech investment that we see with companies that have shown successful clinical stage data being acquisition targets left and right. And especially for some of these large pharma that are trying to move more into the oncology space, Gilead's been historically very focused on virology, so they've tried with Trodelvy to get into the oncology space. Another program like this would make a lot of sense. So excited to see how this plays out, and hopefully it will keep working, give patients with cancer some new treatment options there.
Yeah, and Gilead's not the only one. Evidently, looks like BMS is partnering with them as well. Both are looking at solid tumor treatment, and so definitely a hot area, like you mentioned.
Yeah, I mean, I know ASH was this past weekend. So ASH is the American Society of Hematology, and they focus on a lot of blood-based cancers. So your AMLs and ALLs and things like this. So definitely solid tumors have always been the next frontier, and ADCs have a lot of promise for those types of things where, again, that the fact that it's an antibody, a linker, and a payload, that payload portion of this can make this a lot more effective, or so the theory goes, for solid tumors. So definitely a lot of hope and a lot of investment around that area. And then the other story, also related to ASH, is the data from Insight on their new treatment, Monjuvi. And this is for follicular lymphoma. And basically, they saw a total of about 57% reduction in disease progression and 45% of the patients had already received 2 lines of therapy. So towards your statement on the draft guidance and the standard of care, basically these are patients that have already failed several arms of the U.S. standard of care are going on to the next stage. And this treatment is showing the ability to potentially significantly help those patients.
Right, we're talking about relapsed refractory, and so this treatment essentially 22.4 months progression-free survival versus only 13.9 for the control. So close to doubling the survival, which it's huge. Obviously, it was tested in combo with 2 other therapies. One was the BMS's Revlimid, which is a thalidomide analog, and Roche's Rituxan, which is a CD20 specific monoclonal antibody. And so that's where you were seeing the reduction. And so this specific therapy is a anti-CD19 monoclonal antibody, essentially targets rapidly dividing cells. And so, yeah, it's good to see the success here.
Yeah, indeed. So, in a lot of these conferences like ASH, it's you'll always see the newest things come out. So between that and JP Morgan, it's sort of bellwethers for what 2025 is going to look like. And so far, I'm seeing a lot of positive news. So hopefully that continues.
Mm-hmm.
And of course, before we get to 2025, in terms of KCAS news, we do have a number of holidays coming up. So I know we just had the Thanksgiving holiday, but looking forward to Christmas and Christmas Eve here at KCAS, as well as New Year's Day.
Yeah, it falls right in the middle of the week, too. It's funny. I know a lot of times people would try to schedule vacations when it's Monday, Tuesday, get a little bit of a long weekend. This kind of makes it, I think a lot of people are interested, or at least you and I had discussed that people would just want to be off for 2 weeks, right? I will say we're not off for 2 weeks, so that's clear. But yeah, definitely getting the time off. And I just realized I've said definitely at least 6 or 7 times. So we'll definitely try to say something different moving forward.
Nice. And I know at the end of the episode, we've got to talk a little bit about some holiday meals and things like that and decorations. But maybe that's a good segue into the paper for today. So today's paper is Fluorophore multimerization as an efficient approach towards bright protein labels.
Yeah, and this is in the European Journal of Organic Chemistry. I will apologize. I am just making sure I get the impact factor there. And so what are your thoughts on this? Okay, before you dive into that, I did get it. 3.26. It was as of 2021 that I have here. Wanted to make sure I got that. Sorry, it's been a busy meeting day, like we were just talking about earlier. But it was very in detail paper, specifically on conjugation, multimerization. And we've got a special guest with us today that has a lot of experience in this area. And so, Brian, I want to hand it off to you and get your thoughts there.
Yeah, and exactly as you say, this is not an area where flow cytometrists are usually comfortable to go, right? We like to think of the dyes attached to antibodies as sort of inanimate objects that we don't have to worry about, that work perfectly all the time. But that is indeed not the case, and this paper goes into both how some of these dyes are created, then how different spectra dyes are created. So with that, we better bring in the experts immediately. And towards that, Kelly, if you could give a little bit more of your background and some of your impressions on the paper, then we can sort of start out the discussion there.
Yeah, thank you so much. I really like how at the top of this paper, it's called Very Important Paper.
Yes, I noticed that too. I love it.
If you print it out, I'm like, that makes me giggle, because to me, it is a really important paper. It may only have an impact factor of 3, but I think that it does this beautiful job at going, literally, it's like these guys are talking about their job, every part of their job, the conjugation, the recombinant protein, the fluorophore conjugation. So how many of us get to write papers just about what we do? So did you want me to give a little intro about myself?
Yeah, if you could, that'd be great.
Okay, great. So yeah, Kelly Lundsten. I started in neuroscience, actually. Did a lot of microscopy early on in my career, and came to this pivotal point in my life where I decided not to get my PhD. I didn't see myself along an academic path. And so, just over the course of time, had gotten some training in flow cytometry when I needed to figure out what to do with the rest of my life. I applied for a job at Molecular Probes, which is how I became what a lot of people like to call me, which is the fluorophore girl. I feel like I can accept that title. I don't know a lot of other people giving great training around fluorescent molecules and flow cytometry. As you said, it's not the most comfortable topic for most.
Now, Kelly, before you move on that, I'm curious. So has that title gone through iterations, or was that the first one and it stuck?
Oh, I just remember it was after a talk, somebody had come up to him and been like, "Oh, you're the fluorophore girl." And I just told that story to friends, and they're like, "Yeah, that's consistent." And I mean, I'm a woman now, so maybe we could change that to be a little more appropriate to age. But outside of that, it remains true. A lot of people may not even know Molecular Probes anymore, but they were the ones who originally, discovered and invented a lot of the fluorophores that we base all of our technologies on. Mm-hmm. And I stayed there for about 7 years through an acquisition by Invitrogen. I was an application scientist, just training people. I would give 2 talks a day, almost every single day of the week, trying to help people understand how their tools worked. Because in my view, if you understand how your fluorophores work, then you understand how to troubleshoot them and how to effectively use them. Then I went to BioLegend for about 11 years, and that was varied. That was like a wide range of things that I did, but I did a lot of R&D activities, and I was involved in the development of the Brilliant Violet dyes from their inception through their release. A lot of the dyes that you're familiar with, the spark dyes, the fire dyes, the dazzle dyes. Mm-hmm. We went apparently with a fire theme for being in Southern California. A lot of those are floors that I developed. And then after leaving them, I have my own consulting company. I work for FluoroFinder and Beckman Coulter, a short stint at Thermo. But, I've always been doing the same thing, which is helping people understand their tools. I'm not a chemistry expert. I'm not a real chemist. I like to say that I play one on TV. I certainly play one, at the regional flow cytometry meetings, training people. But, it's not the same as these guys in this paper. They are truly creating the chemical technologies, and I'm helping to adapt those for, cellular analysis and kind of giving guidance on that. So now, we all play our part, right?
We do. And I'm very curious. So the distinction between what you were doing in developing the fluorophores versus what you mentioned about the people in the paper doing, I think, our listeners probably have a background very similar to what Brian had mentioned, he and I, and where, we're aware of the dyes, right, and the differences and how to use them, so on and so forth. But I think very much less so is in that development. And so where do you draw the line? What is the difference between what it was that you were doing versus the authors of the paper?
Yeah, that's an excellent question. I can give you a great example of that. When we were first pitched about the brilliant violet dyes, it was by Brent Gaylord. He used to be with, or he's with Sirigen, now BD. And when he came to pitch us, these chemists, they're so excited about their water solubility and they're so excited about their brightness, but they weren't considering what it meant to conjugate that to an antibody and actually use it in cell analysis. And so that's the kind of bridge. I go to a lot of chemical meetings, and just to kind of see what people are playing with. I do call it playing. I think chemists like to play with their chemistry quite a bit. Just kind of making mutations, modifications, seeing what happens. A lot of times they're looking for collaborations in biology to see if there's an application for this thing they're playing with. And so when it came to the brilliant violets, it was probably 4 times larger than what it is now. They were so excited about their water solubility. It was just such a funny conversation to go back and forth, and it required many iterations before we got to the molecule that would become brilliant violet 421. But it wasn't that we invented that molecule. It was already under patent by Sirigen. They did the work, they did the chemistry. It was a Nobel Prize, platform. But we have to sculpt that into, a tool. And that's what we do in development. I make that distinction between research and development. Those are 2 very different things.
And I think that's always a good point, right? And we run into the same issue just from a philosophical perspective of flow cytometry. Like, what can we do with flow cytometry? We get so excited that we can do like 40 colors and dimensionality reduction, all these things. And then we bring it to things like we were talking about with the investment news on, all right, what does that mean for patients with follicular lymphoma? Absolutely. And it becomes difficult to draw those lines. So, from the flow cytometry perspective, from a dye, we look for something that can be attached to an antibody, as you say, that won't disrupt the sort of binding mechanics of that antibody, that is stable in a variety of solutions that we might need for the staining process, and is photostable through general lab use with some degree of care, ideally at both room temperature and, to some extent, refrigerated temperatures. And these, I think, are the primary considerations of how we're using dyes for flow cytometry, or what we look for in a good dye, between that and the ideal spectra, probably, i.e., can it get excited by an available laser, and is that emission spectra then detectable by the detection technology that we have available to us?
Yeah,
and are we looking at it? Go ahead, Kelly.
To that, I would also add, we didn't even know how bad nonspecific binding could be until we started developing brighter dyes.
Fair.
And so, that element of it, and I think I can't reinforce enough how important spectral stability is going to be. It was one of the things that you had just mentioned, but previous generations of fluors, they were generally sufficient when all you were doing was monitoring a single channel. And one of the first things that we needed to do with the onset of spectral cytometry was pay care to every channel that represented that spectrum. So it wasn't just about, that the increase or decrease or the kind of photo bleaching or the, that loss of intensity, but to the integrity of the entire spectrum over time. And that's something that we definitely are experiencing the ramifications of that all the time. And then kind of third, one thing we weren't considering is something I talk about a lot in my talks. When we were developing the brilliant violet dyes, for example, we were just trying to displace Q dots because Q dots were so problematic and nanocrystals were problematic. We weren't thinking about how they played together in the sandbox. Was one of them beating each other over the head with a shovel, which is sometimes what it felt like when they're put into a big multicolor assay. How do they play together? It was about community. The fluors need to participate in the assay equally without. So one of the things we didn't do at the time was offset their emission. And I think that's also something you're seeing with a lot of the new probes is a better, kind of enabling them to play better in the sandbox together.
Okay. And, you did touch on a question that we frequently get from sponsors as well. what is more important for spectral flow cytometry, the spectra of the dyes or the brightness of them?
You can't say that it's one or the other.
Okay, all right. And that's always my response. I want to make sure we're all aligned here. So that is, yeah. All right. Okay, good.
Come on, Brian. Which is more important, the tires or the wheel on the car? I mean,
yeah. Well, and also we know that not all things need to be super bright, right? That's not going to help us if we're constantly saturating channels, with high abundance. But I mean, that doesn't tend to be our biggest issue with antibody-based detection. It can be a big issue maybe with expression proteins or, that sort of thing. You can quickly go to a saturating level. But we don't tend to have that. We can titrate our antibodies, right? We can use other methods of controlling overall brightness. And the fact is that FITC is one of the dimmest fluors that exists, and yet still one of our most useful. We won't be displacing FITC anytime soon. So brightness, I also find, just from working with FluoroFinder and making a panel development tool that's automated, the thing that we can give on the most is brightness, because all we use are these gross steps of low, medium, and high level antigen abundance. Mm-hmm. And that is, very unrefined and very subjective. But since I think that we are adapting to a future where spectral is king, the quality of the fluorophores has taken on a new life. When we talk about all of these characteristics of new dyes and whether or not we really need them, do I really need to replace ECD or do I really need to replace PerCP 5.5? The answer isn't always going to be absolutely. That's not a black or white thing. But for where we're going as a reproducible assay, yeah, it is. We need to adapt to the new technologies because they're going to be more stable and they're going to be easier to tune and they're going to play better.
Okay.
So one of the things that you had mentioned, and is kind of, I guess, from when I was starting out in Flow, the Q dots, right? And wanted to replace them. But then with Spectral, we've seen this kind of renaissance of Q dots being more useful. I'm curious as to, you obviously been very tied to that, your thoughts there and where you see that going.
Oh gosh, I've got so many good stories about Q dots. That was such an unfortunate product launch. I was there for all of that. Like a joke I tell when I give a talk is that my very last year was almost every single week going to labs to do Q dot conjugations on bench with people. Mm-hmm. And that was when I hit my limit, It is not an easy conjugation. It is not an easy purification. And in all reality, not to get political at all, but it isn't always about whether or not a technology is awesome. It is also about how easy it is to license those mm-hmm. molecules. As we make advances, every company is trying to protect their intellectual property and their market position by either making it easy or difficult to license. And at the time, QDOT was the only way to get to 18 colors. And, Invitrogen very much made a decision that they wanted to protect that by not out licensing QDOTs. So if any other company like BioLegend was up and coming, didn't have a great number of fluorophores available, right? They just had what everybody else had. You have to, I love displacing technologies where they're becoming intransigent with their, protecting technology. It doesn't help us grow as a field. So, with the QDOTs, oh gosh, they're just, it isn't that, I would say this all the time, like, don't believe that they will be failures forever, because that isn't true. There's carbon dots, there's all sorts of nanoparticles that might become very interesting when we can start to make them smaller and more soluble, where the diameter of the core is not the thing that tunes the wavelength of excitation and emission. There's lots of different matrices where you can dope the core with different elements that will tune the spectrum, especially carbon dots and graphene particles and things like that. So I don't think that we should ever think of things as truly good or bad, because that's not real, right? It's just what stage in development, how close is it to something we can actually employ in cell analysis. and hopefully continue to put pressure on other technologies to be better. Like it was also not helpful for us to sit with all the problems that the brilliant violets had. They non-specifically bound each other pretty badly.
Mm-hmm.
But it doesn't mean they're not useful. We use them with a buffer. Is it ideal to have a buffer? No, of course not. We don't want to use a buffer, but it means we don't have to use Q dots. So yay for that. When I was feeling particularly defensive some days, I was like, that's cool. You can go back to using Q dots, just to be a bit of a jerk. But even with the brilliant, polymers, PEG is now this primary component of our cell analysis. Mm-hmm. And that's also something that's brand new. We need to keep these fluors soluble, and the bigger that we make them, the more important it is, the more likely they are to not be as stable in the aqueous environment that is our biology. Mm-hmm. And that has been multiple fold since the brilliant violets, and will be with also the real blues and the real yellows and a lot of the new. Even that paper from how the biobrights are made. It was the second paper by these guys just this year. It's on a PEG scaffolding. Well, when we got vaccinated, all of us became, we had anti-PEG antibodies because PEG is everywhere. It's in my makeup. It's in anything that's miscible. So I think one of the things that needs to potentially change in the future is to wean us off a dependence on PEG and try to find other ways of solubilizing fluorophores. So getting a bit into the weeds of the chemistry, but we did see an impact of that during COVID.
And then if we do that with dyes, will we see the same with another medication?
There's a lot of funny stories, but while we were developing the brilliant violet dyes, we had, like a lot of these companies, we would take samples from donors in-house. So the 20-year-old RAs, or 21-year-old RAs, they would donate blood, get, $10 for it or something like that. And that's the, blood that we would use for testing that day. And we would regularly have people who couldn't be used to test the brilliant violets. At the time, we didn't know why. They would just go on like a not able to use list. And the likelihood is that, once we realized that maybe it was anti-PEG antibodies because of a massive event where everybody was kind of, more people than were testing positive or were having non-specific binding issues, that, kind of gave a path to why, some clarity as to why those original people couldn't be used.
The college bias strikes again. It's just like with all these psychology studies that can, tell you with some degree of certainty exactly how college freshmen and sophomores feel about different things or react to different things. But then when you apply it to the real world, it may or may not be slightly different, right? So same thing, I guess, with their blood and antibodies, that we should expect to see the same biases.
Well, that must have been an expensive study, too, because of all the additional blood draws. You keep on losing students or the availability of samples because of the prevalence of anti-PEG antibodies there.
Yeah, I actually, by that point, I had already left BioLegend. I had left at the beginning of 2020. And it was maybe like 6 months later that they issued, just a notice to people and gave a list of the different fluors that people might see non-specific binding. And, so there's a company called Biodium. I don't know if you're familiar with them. One of the strategies that they actually talk about in this paper is to actually pegylate individual synthetic organic dyes in order to be able to conjugate more of them to a limited space like an antibody. It's actually a patent protected technology of Biodium. And that means that a lot of our tandems, all of us buy fluorophores from Biodium for that reason, that they just make better protein tandems. The acceptors do, acceptors that we buy from them. But then that means that now those protein-based tandems have PEG. So it's wider than you would think of for just the polymers, that dependence. We've applied it broadly,
Okay. So in terms of other sort of pitfalls of new dyes, definitely there's, of course, the peg. There's just general, do they play nicely with others as a consideration? Should we add in there then, I guess, manufacturing base or diversity? As we saw with COVID, that can become an issue quickly if everybody's buying pipette tips from the same place, and then all of a sudden there's no more pipette tips.
Oh, absolutely.
Do we have to worry about with that with dyes?
3 months.
Okay.
There are some manufacturing issues with dyes. I think the one that I can think of that's most prescient was all of the oligos, that kind of the demand on oligos during COVID for testing kits for PCR made it really difficult for what's the floor from Thermo Fisher, the DNA-based dyes, the NovaFluors. I'm sorry,
Yes.
That's the part right there. The NovaFluors, they're all these, these pieces of DNA that are assembled via DNA origami, and I don't think anybody could probably make a case for why manufacturing of NovaFluors should take precedent over PCR probes during a time like that. So there can be some issues in scaling and just demands that can make it difficult for floors. It's absolutely a concern when we're developing them. How reliable, what is the risk assessment for that stage of product development as to how stable that process can be. Mm-hmm. But we never, and also the cost. When we were first making the brilliant violets, they actually were extremely expensive to make. Sometimes you have to go with a contract organization for your manufacturing, a CRM, which adds cost to an already expensive material cost. Always trying to find ways of leaning a process, can help.
Okay. And sorry, go for it, Adam.
I was to say, really interested in understanding in that developmental process. One of the things that we talk about a lot is cocktail prep and trying to simplify that. As you imagine, when you're doing an assay and we're talking about many colors, obviously the cocktail prep is an important part of it. And so considerations for the dyes being amenable to sorts of different operations in cocktail prep to simplify that, standardize it, maybe even automate it. What is your experience there?
Yeah, absolutely. Actually, this paper did a pretty good job at describing the scenarios where polymers can aggregate or mm-hmm. can non-specifically bind. That's been the biggest limitation since we're so reliant on brilliant UV and brilliant violet dyes. Has been their non-specific binding to one another. Mm-hmm. And that, I definitely remember when that first came to my attention. I remember the heart attack that I had because the person who told me, the evidence that they were seeing of non-specific binding, I couldn't believe that I hadn't caught it. Mm-hmm. And I immediately went and tested 2 by 2 every single brilliant violet by each other. That was the biggest limitation to cocktailing. And that was very unfortunate. I mean, it takes us so long to prepare all of our controls and is so prohibitory if we can't cocktail properly. Mm-hmm. So that's definitely something that we need. And I think that will be one of the advantages of the real yellows and real blues is theoretically, I have not tried this yet. I have not tried to dry them down. I haven't tried to do a long-term cocktail. But from what I understand from their marketing is that it should be much better in that regard. And that's going to help us a lot. If we can dry down reagents, it will make, getting to 30 plus parameters so much easier because then we can think about the format that we're offering the reagents and trying to make it easier for people to, it's from an R&D perspective, you have to think about what it's like to be the person sitting there having to dispel tip after tip after tip, going through thousands of tips.
Yeah.
Hurting themselves in their tennis elbow. If there's a different way that we can format how to actually apply the reagents, and if we can dry them down, potentially make them more shelf stable, easier to ship, easier to deal with, under non-ideal conditions, that's definitely super important. And I think that that's part of what the future is.
Ensuring consistency, right? Yeah.
Yeah, we can't just keep going through 30 reagents, 100 tests. I can't, just the amount that we're consuming because we're coming from a lower parameter experience, it can't stay that way. We have to think about what it means to be the person doing the assay.
And so the two options that I see now for exactly that concern, right, are maybe three then. Automate it so that a robot's doing it instead of a person. B, find some of these dyes that do have, slightly better cocktailing capabilities and sort of do it manually with better cocktailing at the bench, or, as you say, have sort of pre-made cocktails that can be dried down and shipped different places. So based on current state of the field, what's your take? Should I buy a robot now, or should I wait for a year or two?
Oh gosh. So I've been doing some work for Beckman Coulter, and I look at their liquid handling instruments with such lust. I'm just like, how do I get one of these in my garage? I would be so much more effective. But then to me, the question would be to put pressure on companies like Beckman Coulter that have these technologies to say, how do you get me one of those that actually meets my needs as an academic lab? How can you scale this down so that you're simply making it easier and more reproducible for anyone to do their research, not just large clinical pharma, applications? So I think that that's an excellent question, and I would like to be the first to buy one of those.
All right, so if Beckman Coulter is listening, and Christmas is coming up, and Kelly would care for a liquid handling robot.
Absolutely.
Yeah, it's been on the list for a while too, right? Well, I shouldn't say it's been on the list for a while, but we've discussed this, and certainly something in the future that we'd like to add to our tools.
Definitely. I mean, Adam, the promise was 1 to 2 new cytometers, not necessarily 1 to 2 new robots, but we'll see what next year brings. Who knows?
Yeah. I actually think that, at this stage, fluorophore development has hit its prime. We are kind of, there's a lot of different ways that we can use fluorescent or non-fluorescent backbones, and once we got into multimers and polymers, I feel like that's going to be this kind of this very stable place where there's going to be small developments, but nothing earth-shattering at this stage. We need to figure out how to diversify the repertoire of what we have. But there are so many other things that are interesting in this space, like using autofluorescence as a parameter and making it, again, easier for that individual at the bench to accomplish their work. There's going to be so many offshoots where spectral is putting a pressure on those things that should help create new opportunity and new business ventures, I think.
I really liked your idea there, the autofluorescence as a parameter. It's almost like the QDOTs thing in a way, because, for a while it was just an interference. It's a problem. Then all of a sudden you get spectral, you're able to subtract it out. And, now you're saying about utilizing autofluorescence as a parameter. So it's certainly evolving.
I honestly believe that is going to be the most important thing. Could you imagine? I'm not positive that this would ever happen, but could you imagine if we actually had an array of different lasers that actually their only purpose was to pull out specific single source autofluorescence, signatures from spectral,
right. What kind of information you could glean from that? Obviously, it's lower cost there. You're not adding antibodies. You would need a machine, to your point, specifically to do that. But you're not making your cocktail preps, and or at least if you're just looking at the different ways the cells have autofluorescence.
Yeah, kind of back to your autofluorescence, or sorry, your cocktailing comment as well. So when I first showed up at BioLegend, everybody had kind of come from BD already. They were just scaling up antibody production. They'd been in business for about 10 years, and they were really focused on clone generation and not much else at that stage. It takes a long time to become what BioLegend is today. And I remember looking at our portfolio of cocktails for lineage, just old school, every lineage marker in PE, and they just weren't selling anymore. Once we got towards 18 colors, they were poorly performing SKUs. And now that I'm part of a consortium called SoulCap, it's also, as you had talked about previously and during the news segment about kind of standardizing and the importance of not just throwing pebbles into the ocean. SolCap is an effort to standardize flow cytometry. And now that I'm more involved in the marker annotation effort there, I wish that we had more intelligent dump cocktails. There's a lot of things that we don't take the time to exclude that could really clean up our data and make it more meaningful in terms of a downstream functional consistency. But because they became irrelevant, as we got into a higher parameter when we could see more things. And so we kind of stopped dumping things, as many things together. I think that there is this, we have to think about what does it mean? How can we help guide people towards better consistency by actually just having a more meaningful cocktail of things to even remove from the assay?
Okay, so better dump channels instead. So yeah, I mean, definitely a label-free assay would be wonderful. I think, I want to say, ThinkSight has what they call ghost cytometry, where they're using essentially confocal microscopy, if I understand correctly, to do something similar to that.
Waveform. I think a lot of them are waveform.
Correct, indeed. Exactly that. And then you can basically look at cells, but my concern is always how well does that work in the heterogeneous populations that we see out of patient whole blood samples as well. It works great for homogeneous cell line populations or even cell therapies where you're growing up a large number of the same type of things and isolating them. But if you have a really mixed population, I'm not sure about how well that would play.
Yeah, I completely agree with you. I've seen a lot of the talks you've seen probably about label-free. I think the proof is still in the pudding on that one, especially if we want to apply AI to that and think that we can pull out, specific health states, for example. I mean, right now, if we can't resolve an activated T cell from an inactivated on scatter, so unless there's something that's extremely distinctive on waveform, we're not talking about a 3D analysis of that cell. That's still a 2D, planar capture of depth of a cell. That can be complicating. There's a lot to be said for depth and volume. So yeah, I'm not sure how I feel about label free yet. I mean, you can call me a skeptic on most things. I think that's why we make good scientists.
Mm-hmm. Well, you have to see it work, right? You said the proof is in the pudding, and we need that data to be able to make those informed decisions.
Well, not to mention the fact, of course, Kelly, as we talked about earlier, if you are indeed now the fluorophore woman, if we go entirely label free, then it's a concern. Like, there's redefinition.
I'm worried about my job.
Right.
Anytime soon. We're still using PE, and I'm pretty sure they found that well before I was born. So
yeah, indeed. And towards that point, I think we've covered a lot of our sort of general questions. Towards your point on basically we shouldn't dismiss things out of hand, like even with non-specific bindings from brilliant violets, or even with sort of difficult spectra or difficult maintenance with the PerCPs. Basically, there's nothing that we need to run away wholesale from as a field, but you would expect basically as we transition more and more to spectral that some of these dyes that don't play as well with others will likely get left behind.
I think the only exception I might make to that is PerCP Sci 5.5.
Okay.
I will give you permission right now to no longer use that fluor at all.
Okay. Run away as a field.
Yes.
Got it.
Very much agreed on that one. It was funny when I was learning a higher level flow cytometry. That was the first one that a person who was teaching me had mentioned, stay away from this one. I'm like, all right, good, I got it.
The way that I always try to teach people about PerCP is you can imagine that it's the castle that needs to be surrounded by the moat. And the moat is the Sci 5.5 fluorophores. And they'll take the assault of the reactive oxygen species in order to protect the PerCP from a quick degradative effect of photobleaching. But it still just leaves PerCP in the end. There's a reason why those two things emit so close together. They weren't designed to be a real tandem, and they're definitely problematic for spectral.
Nice. I had not heard the castle analogy before, but yeah, that makes it much more approachable than organic chem in general, right? So, I guess that's the other question. So going back to the paper itself, again, we covered a lot of the concepts that were discussed in the paper, just in the discussion, but anything that you want to call out about either this paper or the other paper by the same authors?
Not anything in particular. I mean, they really went over just different nucleophilic substitution chemistries to cross-link. How do we get fluorophores onto the antibodies? I know that, now that, say, Biorad has the super brights coming out, they're trying to find more mix and match, more accessible ways of people being able to conjugate their own antibodies with large polymers. Some of that was, pretty standard. I have to honestly say, over the course of my career, I haven't seen nucleophilic substitution have any creativity to it. You just have to have a site to conjugate. You can only get it so many ways. If anybody's curious, there's the structure of the brilliant violet in there, the polyfluorine. I love this paper, and I've told them it's in my talks a lot, this citation, because it's just really important. They do this great job of walking you through what they're thinking and what they have access to and what they understand to do, to develop their own technologies. And I find it to just be very educational.
Agreed. And as you say, not so in the weeds with the organic chemistry that tired flow cytometers can't also get some value out of it as well. So that is always good. Agreed. Adam, any other thoughts on the paper or on the sort of fluorophore question in general?
No, I think we've covered everything there. I am kind of curious, Kelly. We talked about your least favorite fluorophore there, and rapidly getting rid of Percy-P Sci 5.5. Thoughts on a favorite, or even just a favorite story around the fluorophore?
Oh gosh, that's like my whole career right there. I mean, I'm always going to say that BB421 is my favorite. I mean, if you have ever seen me in person, I have a tattoo on my forearm of all of the fluorophores that create the rainbow.
That's awesome. Because
I find that grad students love to be regaled with why the chemistries are different, and I can quickly show them off of, my demonstrative tool on my arm. But I mean, BB421 is going to be my favorite, but I am extremely impressed with the real blues and the real yellows. Mm-hmm. Whether or not that's going to be something that's open to continued development, anytime something is newly released, like the brilliant violets were, there needs to be a generation of things that comes after it that's better. And I think that that's what we're looking at right now. And so that's really exciting to me.
Nice. I will take the opportunity to inject a quick pun that's been on my mind for quite some time, related to rainbows and tattoos and things like this. So the question is, Kelly, where do bad rainbows go?
Into the blackness?
Close. They go to prism. It's a light sentence, but it gives them time to reflect.
Oh no.
Sorry, I had to. But thank you. And I had not thought of tattooing things on there for presentation purposes, but I will add that to the marketing team's list of things, though, because that will be potentially useful.
I do know a tattoo artist, so you just let me know.
That is it.
When you're a lifelong teacher, you have no idea how many times it comes in handy.
Excellent. All right. So I guess from there we'll proceed into a quick request for feedback from our fans. This fan feedback request doesn't make sense, so I'll make another one up on the fly. Apologies here, but I'll ask our fans the same question that we asked Kelly. What are your favorite or least favorite fluorophores? So if you can reply in comments, then we'll make sure to feature it on our next episode. And then also in terms of just moving on with our colorful theme here, going into Christmas decorations or holiday decorations in general, do you guys lean towards single color or multicolor decorations?
Kelly, I'm going to guess for you, you've got to be multicolored, no?
Actually not. I grew up in Pennsylvania, and I have a Moravian star at the center of my window, and it's this warm white. And I love when people pass. I hope that they feel happier by seeing something so pretty. And so I have a very warm white theme all around my house.
Nice.
I like that. Yeah, it is definitely decorations like that are very pretty. I'll say for myself, I had long grown up being the kind of multicolor, but my fake tree, which I'm not happy that we have a fake tree. I've always grown up with real trees, but I lost that battle. And so it has a switch where it can go from just your single color white light light to multicolor. And so I actually click that one rather than keeping it on multicolor or white. So I get both.
So my question for you, Adam, is does it blink?
It does. It blinks back and forth between the colored lights to the white lights and back and forth.
Very nice.
How about you, Brian?
Yeah, I personally also prefer the white in most cases, but my 7-year-old has decided that his definition of beauty is as many colors as humanly possible. So, if he sees like a 2-color thing, he's like, it's kind of pretty. But then if he sees like a 20-color thing, he's like, that is awesome. So,
I like him. So he takes after his dad then, huh?
Indeed. Yeah. So his our Christmas decorations are indeed very multicolored at this point. Yeah. And then, also going into the holiday season, any interesting gift, either philosophies or procedures there?
Oh gosh, I love white elephants. I absolutely love them. When I was with BioLegend, I was traveling quite a bit, and you guys know how it is when you're on the road a lot, you get sick, So as my white elephant exchange, I brought a box of tissues and those stuffies that are different viruses. I was so proud of myself. And I think like cough lozenges. That was my white elephant contribution, was the little plushie and then like a kit for cold, recovery. And nobody thought it was nearly as funny as I did. I was so amused with myself.
I really was thinking you were saying that the gift was getting everybody else sick when you came home at first. Listen, guys, you're going to be off for a week. See, that's my gift to you.
Immune system strengthener, all those things, right?
All the positive benefits you could get from that. So it's interesting, though, with these kind of gift things, I feel like they evolve through time with families, right? You start out when everyone's little where it's just like a ton of presents everywhere. But then as the family grows, you realize that's really just not something that you can keep up with. It becomes very expensive. And so, the white elephants, the exchange become the thing. So I started something with my siblings where we basically had names in a hat, didn't know who got who. And then everybody ordered a present and sent it to the person who they had. And we then did a Teams thing since we're all in different locations, kind of far flung. And you had to guess who it was that sent you that gift. And it was fun. What the thought process was leading up to who your guess was. And so now we've expanded that out to the nieces and nephews and everybody. So looking forward to how that evolves.
Nice. And my favorite gift exchange philosophy is definitely the Yankee swap, where there is an element of both chance and stealing and things like that for the holiday season. Always, one of my favorite Office episodes was, of course, the Yankee swap, and the more that we incorporate that, the better, I think. So adds an element of flavor to it.
Now, do you have a limit on how many times a gift can be stolen?
Once.
Just once, okay.
Indeed. Yeah, otherwise then it's the one thing that everybody wants, and then you have to constantly swap the one thing, and then indeed. But if you limit it just one time, then it makes for relatively even encounters there.
Mm-hmm, mm-hmm. Nice.
Yeah. All right, excellent. Well, again, I want to thank Kelly for joining and for talking through some fantastic fluorophores on our show today.
Thank you so much for having me.
Yeah.
Kelly, it was really a blast. Enjoyed getting to meet you on our podcast and also hearing your stories. It was a lot of fun. Thank you.
Great. Thank you.
Yeah. And for our next episode, we're looking at predictions for the biggest changes in cell therapy in 2025. So we'll be looking forward to episode 16 there.
Yeah, it's been a big topic for us, cell therapy, and obviously for all the reasons, right? It's just big out in the industry. So looking forward to really diving in there.
Absolutely. All right. Well, thanks all for listening, and we'll talk to you next time.
Thanks, everyone. Take care.